Welcome to the lecture on different techniques in polymerase chain reaction, PCR. PCR is a crucial tool used in molecular biology to amplify specific DNA sequences. We will discuss 12 PCR variants, each with distinctive characteristics and applications. Let's explore the fascinating world of PCR together. Real-time PCR, or quantitative PCR, amplifies and measures DNA in each cycle using fluorescent probes or dyes. It is sensitive, accurate, and applicable to gene expression, pathogen detection, and genotyping. Reverse transcription PCR, RT-PCR, is a variation of PCR used to amplify RNA sequences. In this technique, RNA is first converted into complementary DNA, cDNA, using an enzyme called reverse transcriptase. The cDNA is then amplified using traditional PCR methods. RT-PCR has revolutionized gene expression studies as it allows researchers to examine the levels of specific RNA molecules in a sample. It is commonly used in studying gene regulation, RNA virus detection, and cancer research. Nested PCR improves the specificity and sensitivity of DNA amplification by using 2 rounds of amplification. External primers are used in the first round, followed by internal primers in the second round. This method is useful for amplifying low-abundance targets like viral or ancient DNA. Multiplex PCR allows for the simultaneous amplification of multiple target DNA sequences within a single reaction. It achieves this by using multiple sets of primers, each specific to a different DNA sequence of interest. Multiplex PCR is utilized in various applications, including genotyping, microbial identification, and forensic DNA analysis. It offers advantages such as reduced sample consumption, cost-effectiveness, and faster turnaround time. However, careful optimization is required to avoid primer interference and ensure accurate results. Hot start PCR is a technique that minimizes nonspecific amplification and improves PCR specificity by preventing the initiation of DNA amplification at room temperature. This is achieved by incorporating a modified DNA polymerase that remains inactive until elevated temperatures denature the inhibitory antibody or aptamer that blocks its activity. Hot start PCR is particularly useful when amplifying complex genomic DNA samples or when dealing with low abundance targets, thus enhancing the amplification efficiency and sensitivity. Touchdown PCR combines specific and nonspecific temperature cycling. It starts with a high annealing temperature for specific primer binding, then gradually reduces the temperature. This improves efficiency, specificity, and is useful for amplifying complex DNA targets while minimizing nonspecific amplification. Digital PCR is a technique used to accurately count DNA molecules. It works by dividing the reaction mixture into many small chambers, amplifying the DNA, and then determining how many chambers contain the DNA. It provides precise and sensitive quantification without external standards. Colony PCR is a technique used to screen bacterial or yeast colonies for the presence or absence of a specific DNA sequence of interest. It involves directly picking a single colony from a culture plate and adding it to the PCR reaction mixture for amplification. This allows quick and efficient identification of colonies containing the desired DNA molecule. Colony PCR is commonly used in cloning, genotyping, and microbial identification studies. Multiplex ligation-dependent probe amplification, MLPA, is a PCR-based method used to detect copy number variations, CNVs, or deletions/duplications in specific DNA sequences. It utilizes multiple pairs of target-specific probes that hybridize to their respective complementary sequences in the sample DNA. Ligation of the probes followed by PCR amplification allows for accurate quantification of target sequences. MLPA is widely used in genetic diagnostics, identifying disease-associated CNVs, and cancer research. Allele-Specific PCR is a technique used to detect specific single nucleotide polymorphisms, SNPs, or point mutations. It involves the use of allele-specific primers that specifically anneal to either the wild-type or mutant allele. By incorporating a mismatch at the 3 terminus of the primer, discrimination between the 2 alleles can be achieved. Allele-specific PCR is commonly utilized in genotyping, genetic disease diagnosis, and pharmacogenomic studies. Inverse PCR is a technique used to amplify DNA sequences when the flanking regions are already known but the internal region is not. It involves the digestion of the DNA sample followed by self-ligation to circularize the DNA. Subsequent PCR amplification using primers designed from the known flanking regions results in the amplification of the unknown internal region. Inverse PCR is useful in applications such as gene cloning, DNA walking, and identification of flanking regions in transposon mutagenesis. Methylation-specific PCR, MSP is a technique used to analyze DNA methylation patterns, which have critical roles in gene regulation and various diseases, including cancer. MSP employs bisulfite treatment of DNA to convert unmethylated cytosine residues to uracil while leaving methylated cytosines unchanged. Specific primers are then designed to amplify either the methylated or unmethylated DNA sequences, allowing for the differentiation between methylated and unmethylated alleles. MSP is widely used in epigenetics research, biomarker discovery, and early cancer detection. To sum up, PCR is vital in molecular biology with many applications. We covered 12 specific PCR techniques in this lecture, each offering unique features and usage. It's crucial for those in the field to understand these techniques for scientific advancement. Keep exploring and utilizing PCR for groundbreaking discoveries.